believed to be an elixir of life, useful for rejuvenation and possibly for achieving immortality. And somehow this latter part is that the scientists want to be recognized even after our death for some significant achievements. And so is there such a philosopher's stone for brain imaging. And at the Martinos Center, of course, we do a lot of multimodal imaging, use different methods in combination. And the reason why we do so is that different methods can answer different questions. And here's this nice citation from Kipling, which says, "I kept 6 honest serving men. They taught me all I know. Their names are what and why and when and how and where and who." And let's look at these methods. This is kind of a selfish picture of the situation. Here at the center is source estimate based on MEG in this case, and this is an auditory evoked response. And you can see in this movie that on the inflated cortex, let's see when it returns, you start seeing activity at 50 milliseconds after the onset of the tone here, and then there's the N100 response following. So this movie is based on MEG and anatomical information about the cortex, which has been obtained with anatomical MRI. And these have been combined together to produce this movie. Then EEG can help us, together with MEG, to give information about sources that are radially oriented with respect to the skull. And finally, fMRI, the hemodynamic response, can give us some additional spatial information, but it must be, of course, remembered that fMRI measures a different thing from MEG and EEG. It measures the hemodynamic response, not the actual postsynaptic currents that we are interested in. And finally, there is optical imaging, which also gives limited amount of information about the hemodynamic signals. We are very used to seeing this picture where there's location, that is spatial resolution, and timing, that is temporal resolution, plotted for each of the imaging methods. MRI, anatomical MRI, which gives us at the very slow temporal scale information about structure and connectivity, that is where something is located in the brain and how these different brain regions are connected to each other. Then there's fMRI, which provides a better temporal resolution and tells about hemodynamic quantities and can tell us which brain areas are activated and to some extent how they are connected. But we need to remember that this connectivity information is the co-fluctuation of the hemodynamic response, and it's not yet very clear what the neurophysiological significance of that is. And finally, there's MEG and EEG, which are less precise spatially than fMRI, but provide much better temporal resolution. That is, they look at electrical activity of neurons, and they can actually answer many questions. Which are the brain areas activated, when they are activated, and how they are connected. And these methods are, of course, all non-invasive and can be at will used in healthy human subjects. But for MRI, really the time is not so relevant, so I wonder what the meaning of this graph is for MRI. And then the same applies to some extent to fMRI, because one could well ask, if there was a method that was spatially precise, would we be interested in the hemodynamic response at all? It might be interesting in some diseased state, but diseased states, but what happened? What happened? But window. This is remarkable. Remarkable. diseased. No, this was a diseased computer. So for the fMRI, we might ask, is the hemodynamic activity really interesting to us at all? And for MEC and EEG, we tend to speak about the good temporal resolution, but we forget that MEC and EEG actually give quite good spatial information also. So it's not so that fMRI and MRI give us the very precise spatial information, just like that, and MEC and EEG give the temporal information. They tell about different things. And I will talk a little bit more about this second sentence later, that is the inverse problem can be made less ill-posed, so that MEC and EEG are by no means ambiguous in terms of the location. So what we learn from this is actually that all these methods have their strengths and weaknesses, which are not necessarily these two axes, but different strengths and weaknesses. And they could be used in clever combinations with each other. And therefore I suggest that this diagram should be abolished, because it leads our thoughts to the wrong path, thinking strictly about this temporal and spatial resolution. So a little bit about MEC and EEG. So as you know, EEG measures the electric potential on the scalp, or also inside the head if we talk about electrocardiogram, and MEG measures the magnetic field outside the head, and these are generated by the same basic mechanism, the postsynaptic currents, and they can, as I told earlier, tell about different aspects of the brain activity, which areas are activated when and how these activities are related to each other. And MEG and EEG have certain benefits, especially MEG has a very subject-friendly measurement setup so that the subjects often tend to even fall asleep in MEG, and MEG has this precise timing and a reasonable spatial resolution, and in fact so does high-density EEG with some limitations. Oops. And it's especially important that we get the spectral content of the signals also from MEG and EEG, which relates to the actual physiology. And in some ways, fMRI is a little bit abstract. It measures these quantities which cannot be related easily to the physical events in the hemodynamic response. But MEG and EEG measure the electric currents very directly, so that we get this very nice quantitative information from MEG and EEG. Which, yes?
So are you going to discuss at some point the issue of subcortical sources for EEG and MEG, because given that you were favoring MEG as a more direct measurement, how do you go about understanding half of the brain which is subcortical?
So I would already now say that it's equally difficult to tell things from EEG as from MEG as the deep sources are concerned, I feel, in the presence of the cortex. If the deep sources would be alone active, the situation would be different. I mentioned the spectral content, and actually, especially in terms of connectivity, whether we have activity in the alpha band or lower in the beta band, or in the higher frequency bands, we may be able to relate these frequencies to connectivity between subcortical structures and the cortex, within the cortex, or locally within the cortex. So therefore these frequency bands can be very useful to know the precise precise frequencies present in the signals. And another reason is that we are starting to be able to make model predictions about MEG source estimates. So what we can do is from one or EEG source estimates for that matter, we can first of all measure non-invasively currents on the cortex using MEG or EEG and get these macroscopic current estimates as a function of time. And then what we can do is to do computational neural modeling, that is construct a biophysical model of thousands of neurons and predict what the macroscopic current would be, what its temporal behavior would be. And then we can kind of make mechanistic predictions of what would cause differences between conditions. And this is what Stephanie Jones has been doing early on at MGH, but now at Brown University. And for example, looking at differences in evoked responses between conditions in which the stimulus has been identical at the threshold of being detectable, and there's a clear difference between the evoked responses to those stimuli which have been detected and those which have not detected, and she was able to relate these changes to specific changes in the connectivity between the neurons. And now, of course, this doesn't by itself prove anything, but then she has consistent evidence in some other conditions, including some the statistical characteristics of rhythmic activity, which also fit into this picture. This is not yet conclusive either. This may be still a happy accident. But now the model has the following property, that it can not only predict the macroscopic currents, but also predict what would be measured invasively from the cortex. And this opens up the possibility that if the system we are looking at is similar enough across species, for example, the primary somatosensory cortex, you could do experiments in rats and actually see whether the model is correct. And then you start getting confidence that your mechanistic explanation in humans is correct because it's actually present in rats in the level that can be precisely predicted by the model. So we start to believe that these models have this property that they are able to do forward predictions. So like tell beforehand what would have been measured. And at least Stephanie, I've noticed, is now having these kinds of insights that I think I know what is going on. So on the basis of her model, so she's able to predict. So this is one direction that MEG and EEG are going to, these explanations model-wise. So notice that this model is used after we have used MEG and or EEG independently to predict the currents, and then we have an explanatory model, rather than having a model which is kind of embedded in the source estimation. That kind of model is more difficult to verify this animal-wise because it's worked into the analysis. So everybody knows, of course, that there's the inverse problem in MEG and EEG, and it's said.
Well, I'm not sure everyone here knows that, so it could be worth spending a little time.
Yes. So the inverse problem means that coming from the measurements to the actual currents in the brain is not a uniquely posed problem. So you cannot estimate the currents in the brain uniquely from the measurements unless you apply some additional constraints. And in addition to this non-uniqueness, there's another facet for the ill-posedness, and that is that the solution may be sensitive to noise. You may change your measurements a little bit, and your solutions change widely. And the latter can be mitigated by regularization, and in general, regularization means that you lose spatial resolution. So these things are linked together in this way. So that if you have a lot of sensitivity to noise, it means that you have to regularize more, and then you lose spatial resolution. And now the obvious solution to this is to make the problem less ill-posed, so that you need to then regularize more, and then you increase the spatial resolution. And one way to make the problem more well-posed is to bring in constraints. For example, you can tell that the sources must be in the cortex or in certain deep structures, or at certain locations in the cortex on the basis of fMRI, if you believe that fMRI and MEG and EEG see the same activity by different means. And another way which has been overlooked for the whole 21st century, I think, is to improve the hardware, to measure closer, and I will talk about that later. But let's show where we are right now in terms of applying all these constraints. So this is an example of a study where we have answered all of these questions: which are the brain areas activated, when, and how they are connected. And this is a study which aimed at looking at connectivity in autism. So on the basis of our results, we can say that both long-range connectivity, that is in the distance scale of centimeters, and local connectivity within a brain region of about a couple of square centimeters in size is impaired in autism. And how we arrived in this conclusion was that we performed an event-related or evoked response study where we compared responses to faces versus houses, and then as a measure of long-range connectivity we took coherence, which is basically frequency-specific correlation. So, and then for local connectivity we looked at phase amplitude coupling, and then asked whether these are abnormal in autistic subjects, and is there any connection between these measures and behavioral data that is collected in the clinic. And the first remarkable observation is that there's no difference between the groups in terms of the evoked responses. So in blue, now we lost the pointer. So in blue we have responses to phases in the fusiform phase area, which is here in the ventral surface of a hemisphere. So this is one hemisphere seen from below. And the yellow is the region which we are looking at, and that's determined on the basis of having significant difference between the houses and faces conditions. So faces being blue in the upper figure and houses red, and dashed lines are autistic subjects, and solid lines are typically developing normal subjects. So there's nothing much different between them in terms of the evoked responses. And then in the lower part, there are different types of faces: angry faces and fearful faces, and again, dashed and solid lines are autistic and normal subjects. So no difference. Is there any difference of connectivity from this area to other parts of the brain? And the answer is yes. So we calculated coherence using this as the seed region. So meaning that we calculated the coherence from the fusiform phase area in the ventral surface of the brain to everybody else, every other region in the brain. And then we compared the phases and houses conditions at different frequency bands and found interesting differences in the alpha band, so that in the normal subjects there was a difference between phases and houses in connectivity from FFA to specifically these three regions, which are precuneus, anterior cingulate cortex, and inferior frontal gyrus. And it's interesting that this contrast didn't exist in the autistic subjects, and it was even reversed. It's weaker but partly reversed in the autistic subjects. So basically this connectivity didn't see any difference between the between the two conditions. So this is the first difference we found. This is kind of a standard connectivity difference that you could determine. So this is basically correlation or coherence difference. Yes?
Sorry, what do the colors represent?
So the red means that for phases the connectivity is stronger than for houses, and for blue that is vice versa. So this is like so the difference connectivity between.
And the connectivity is this the same frequency loss.
So it's basically the correlation in this frequency range. So you can think about the data being filtered to 8 to 12 hertz and then taking just correlation between the signals.
So you found this difference only in the alpha band?
Yes. And specifically at different time points with respect to the stimulus, depending on which region you looked at. So, and it seems that it's just the distance, because this is closest to FFA, and this is probably farthest physically. Yes.
Follow up on this too. In this particular example, when you say the emotional phases are greater than houses in terms of connectivity, what does that actually mean in percentage difference?
So this is C coherence. So this is now compared to the baseline coherence. So
this is the baseline coherence for a given subject.
So this is for the group now.
Okay.
This is for the group. So but this is basically saying that there's a significant difference between the conditions specifically at the different time points as you calculate the coherence. So the coherence is of course calculated across the epochs.
Right. Is there any difference between the severity of the ASD and the degree of?
We will come to that. This is the interesting part. So yes.
Sorry, what does the frequency information tell you about connectivity? Is it a different type of connection?
It's
What does it represent in terms of why didn't you see something in the gamma band?
we didn't see anything in the gamma band. The gamma band connectivity is probably local.
So what's the difference, physical difference? What does the fact that it shows up in alpha band tell you?
I don't really know, but exactly it will tell you physically. But it's in the following it's even more specific, because this is not coherence, but this is phase amplitude coupling, meaning that we find that when alpha is in a certain phase there's more gamma. So the gamma is modulated by the alpha specifically, and specifically at the fusiform phase area. And so this is the modulating frequency, and this is the modulated frequency, if you like. So that this is the baseline modulation, this is the houses modulation, this is the neutral phases and emotional phases. And now it turns out that when you compare the phase amplitude coupling between the emotional phases versus houses, there's a huge difference between this and this in the normal subjects, but there's almost no difference in the autistic group. Now this is not yet clinically nothing more than interesting, because as you pointed out, the question is this related to the severity of the illness in this particular person? And so for this we made a 5-dimensional plot, which is shown here. So this is a 5-dimensional plot, because on the 3 axes we have the coherencies from the fusiform phase area to these 3 regions, which were different. Then in the color we have like the severity of the autism diagnosis, so that red is more autistic basically. And the size of the sphere is the local connectivity, the strength of the phase amplitude coupling between alpha and gamma. And now you see pretty much by eye that even if you don't do any more careful analysis, that small spheres which are close to the origin of this figure are red. So that the autistic subjects show all of these features in an individual level, and then been made more precise analysis across different dimensions. But this is just showing this, trying to show this all together. It's a little bit hopeless to show 5 different things together. But anyhow, this gives you the idea that there's some hope that some rather complicated neuroimaging methods might be predictive in an individual level. So this is kind of an objective diagnosis. It's a very difficult analysis to make in practice, but still the experiment is rather simple. It's an evoked response study, and then a miracle happens, and then you get these 4 numbers which you can relate to the diagnosis. So who knows, maybe something like this will be useful in the future. Now of course, this not only requires that this is predicting something in an individual level, it also has to lead to something. So there has to be something what you can do about this. And that's the next step. So what could you do about this? And maybe understanding that these kinds of things are abnormal might help you also do some treatment which is mitigating these things that you have observed in imaging. So briefly about anatomical MRI, I think the big leap forward in the use of anatomical MRI in multimodal imaging came from when, starting from Anders Dahle and co-workers, people started to produce these geometrical models which provide quantitative information about structures like volumes and shape of the cortex and so forth, so that we can use these anatomical data as constraints in MEG and EEG, and we can map the data on the cortex across subjects and so forth. So this was a big leap forward, not to look at just normal anatomical slices, but these precise geometrical models. And for example, the primary currents that we look at on the cortex in the pyramidal cells are guided by the geometrical arrangement of the pyramidal cells, meaning that the currents are normal to the cortex. And MEG doesn't see the radial currents which are normal to the scalp, and therefore it doesn't see the gyral currents precisely, but EEG does, and both MEG and EEG see currents in the sulci. So that MEG is selectively sensitive to these, but whereas EEG sees both, and therefore is more difficult to interpret. And it must be really emphasized that this geometrical model of the cortex can be done completely automatically now, thanks to the efforts of Anders Dahle, Bruce Fischel, Matti Sereno, and others. So that with the short anatomical MRI scan, you can produce a lot of quantitative information about the brain, which can be used in MEG and EEG modeling. And especially nice is the fact that you can inflate the cortex, like here to see in the sulci, so that you can get this picture of currents in the sulci, which is more difficult to see when the cortex is folded. And also you can continue this inflation process all the way to a sphere, and then align the folding patterns to an average brain, and then map the MEG activity from an individual to an average. And in that way, do group analysis not in volumetric morphing, but morphing which is based on how the patterns of sulci are appearing in the individuals. And this turns out to align things much better than volumetric mapping for cortical activity. And of course, there's other anatomical information that we can obtain, and one such piece of information is the connectivity, which we haven't used so much, but it thanks to the improvement of not only the reconstruction methods of the connectivity, but also of the MRI systems, these pictures start to be more reality than science fiction. They used to be a lot of fiction in them. This was found out so that people scanned monkeys for longer and longer time, and they noticed that these diffusion MRI pictures changed, so that you needed much better quality pictures to really get the details right of the connectivity. And now we are at the point where that starts to happen. Then a little bit about functional MRI. So, yep.
So people at NIH typically will sometimes run a brain for 72 hours over a holiday weekend to get really high resolution.
Mm-hmm.
So is there a way of getting that degree of enhanced resolution with longer time in the scanner? I mean, what humans are not going to sit around to, for more than 20 minutes or an hour or something like that for an imaging paradigm. So how are you suggesting that?
So we solved it so that we got Siemens to build a totally new MRI system, which was designed for this connectivity. Yes, so that it can do higher gradients and do the connectivity much better. And the motivation for this was that, as you say, if you scan for 72 hours, then you can get something that starts to be stable. So the idea is that you can get more of the long monkey scan-like pictures from images from humans with the finite amount of scanning, and this is becoming reality. But still, there is a question which connectivity is correct. I will come to this a little bit later. So functional MRI, functional MRI measures the hemodynamic responses, and since the hemodynamic response is so sluggish, it can answer the when question very imprecisely, and maybe not the right when question at all. And then there's this remarkable observation that there are correlations between the hemodynamic responses across brain areas, and now people are measuring correlations in whatever population they find. This is like repeating the story of event-related fMRI. So, but fMRI has really produced remarkable precise mapping for example, the visual areas in humans, which was earlier on possible to do only tediously in monkeys. And one can even write to the cortex letters like MGH by designing the stimuli properly using the visual magnification factors. And another observation which is also visible here is that if you, with help of the cortical surface reconstruction, in 70 MRI reconstruct the MRI signals on different layers of the cortex, the signals change, as shown here. So these go from the white matter to the pial surface, and you see that the signal pattern changes. And the reason why we believe that this change is real is that it also is related to connectivity changes. If you correlate different layers in two different areas of the brain, you see not a random pattern, but a pattern which might indicate input-output relationships in terms of fibers connecting from deep layers to superficial layers, for example. So then these intrinsic brain networks have really become a popular concept now, and they provide a lot of information about how the brain is organized in terms of these resting state signals. But it's interesting that the 90s story repeats itself in the sense that it seems that the fMRI community hasn't heard that since 20s in EEG these resting state signals have been measured starting from Hans Berger's first EEG study. So this, the human memory is a wonderful thing. So, but how to use MEG and EEG together with fMRI? That's not at all clear. There are all kinds of attempts. This is one attempt we made, but it hasn't become very popular, maybe for the reason that there's no practical software which everybody could use. But this method is called FIRE, and the purpose of it is to use fMRI and MEG and EEG information together. And the basis of the method is that if we parcellate the brain in a suitable way, we can then assume that within each parcel the neural responses are the same in terms of the temporal, the time course, just the amplitudes are different, and the same applies to the hemodynamic response, so that the hemodynamic response could vary across the parcels, but is the same within the parcel. And the purpose of this approach is to account for the fact that sometimes in MEG we have silent sources, and sometimes in MEG and EEG we have silent sources, and sometimes in fMRI which are not visible in the other method. And here we performed a simulation which illustrates how this works. So in this first scenario there are no silent sources, and the sources are 32 milliseconds in location A and in 38 milliseconds in location B. And this is normal so-called minimum norm estimate, which reconstructs the currents on the cortex, and it can approximately tell where the currents are. Then this is fMRI weighted minimum norm estimate, which works well also, and this is sparse source estimates which uses fMRI weight, and that's okay as well in the simulation, as well as is FIRE, except that the FIRE imposes the whole parcel to be activated together. But now the interesting thing is to see what happens when there is fMRI signal, but no MEG-EEG source in this location. So in this case, the normal minimum norm estimate tells the story with pretty precisely, so that you of course get the same as in the previous case, but when the fMRI signal is missing, you don't get any any MEG-EG response anymore, but in the fire method it basically falls back to the normal minimum norm solution in the absence of the fMRI. So if the fMRI is missing something, then we don't use it in any way. It's just the estimate of MEG and based on MEG and EG remains intact compared to the situation where we didn't have the fMRI information at all. So it remains to be seen whether these kinds of methods are actually finally going to be useful, and why I am wondering it is that the larger area, for example, is activated in MEG and EG, the more there's cancellation in MEG and EG signals, because there are different orientations in the currents within this activated area, and therefore, if there are all orientations of current present in an activated area, we see nothing in MEG and EG, and we see a lot in fMRI. So these differences between fMRI and EG and MEG may stem from purely geometrical factors and not from some mystical factors, more mystical factors which relate to the coupling between the hemodynamic response and the electrical activity. So there might be some very basic geometrical factors which contribute to the differences. So this means that in fMRI, really, the observed macroscopic hemodynamic response, which we are after all looking at, is just the scalar sum of the microscopic responses. But in MEG and EG, there is also the vectorial factor in the summation, which complicates things. Now I think I will do the following. I will jump to a few slides forward and come back to the question: can we make MEG and EG better? And so, as we discussed before, there's the ill-posed inverse problem, which is composed of this non-uniqueness and sensitivity to noise. And to get rid of the sensitivity to noise is to bring the sensors closer to the sources. So then the signals will increase and we get more spatial detail, and the contribution of noise decreases. And of course, we should decrease noise at the same time, makes more sensitive sensors. In the present MEG systems, which have existed since the end of last century, the noise level is at best about 2 to 3 femtoteslas per square root of hertz in the white noise range, and there are about 300 sensors using low temperature squids which measure the signals in a helmet-shaped array about 20 millimeters away from the head, 2 centimeters. So this far, and if there's some additional spacing, it's about an inch. And then if you think that the skull and the scalp are a centimeter and the sources are maybe a centimeter into the brain on the cortex, you are 2 inches away. You are so far away. So if you could squeeze away 1 inch, that would be wonderful. And we have tried to do this partly, at least, in a new baby MEG instrument which is installed in Boston Children's Hospital. And this instrument has a small helmet so that it fits babies up to and infants up to 3 years of age. So this phantom here is also a baby-sized phantom. It's about this size phantom inside the baby MEG. And in the baby MEG, we have still on this small helmet the same number of sensors as in the adult. So basically 300 sensors, 275 sensors, and then there are additional sensors for noise cancellation. And the distance from sensors to room temperature is only 7 millimeters. And so we can, with this device, see much more precisely what is happening in the brain. And this means that maybe, first of all, we can achieve a resolution which is about in connectivity, which is about the distance between adjacent sulci on the cortex, and maybe we can also look at individual evoked responses instead of making averages. And because the sensors are denser, we can get more spatial resolution, and because they are closer, we get higher signals so that we can detect with fewer averages. And this is simulated here so that these are the vertical axis. We have localization error on the left and signal noise ratio on the right. For baby head in blue in an adult system and in green in the baby imaging system. And you see that both for temporal and visual signals, the localization error is greatly improved for all current densities in the cortex that we considered, and also the total signal is improved. Signal noise ratio is improved when you go from the adult system to the baby imaging system. So this opens up the interesting possibility that if we have real-time software which we are developing, we could do the following. We could, for example, move a tactile sensory stimulator and look at the activity in the cortex like we do in TMS. We plot the E field in the brain. We would be able to instead plot the actual brain activity for individual stimuli using this new software we have, which would process the data online. But that in babies would require doing something very demanding. That is, first of all, attenuating the noise online, but then determining the head position of the baby online, and then changing the calculations, the forward model calculations and the inverse calculations on the fly as the head moves, so that we could get the brain activity at the right location all the time in the brain. This is a very interesting challenge, and really worth doing with the baby imaging system. Now, let me return to the idea of putting the sensors on the scalp. So this is now an adult head here, and this is the present imagery array. You can see how far away the sensors are. They are. You almost think you couldn't detect anything so far away from the head. So, and on the right we have the localization error of a focal source as a function of location on the cortex. So red is worse and blue is smaller error, and you can see that when you put the sensors close to the scalp and increase their number accordingly, the errors become much smaller. But then it's relevant to us, how about a 500 channel EEG on the scalp? And the surprising answer is that the EEG is not going to be quite that good on the lateral sides of the brain, because the EEG patterns are smeared by the skull and the scalp. So in EEG there's both a distance effect and the smearing effect present, but in MEG there's only a distance effect. And therefore MEG can be vastly improved if the sensors can be put on the scalp, and there's some hope that we can do so. There are, first of all, so-called atomic magnetometers which are becoming very promising to do this type of things. Then there are what is called quantum diamond sensors, which unfortunately are in the microscopic scale at the moment, so there are not macroscopic sensors, but they can be operated at high magnetic fields also. And then there are high DC squeeze operating in liquid nitrogen temperatures, but they seem to be always in the horizon, even though there are some recent promising developments. But if such sensors would be available, we could measure MEG on the scalp, so meaning that we could vastly improve the temporal resolution. And not only that, if the sensors would be like loose electrodes, magnetrodes, we could produce with a 3D printer a helmet for each person. Take an MRI, produce the helmet, which had would have slots for these magnetrodes, put the magnetrodes in, and then we would have an MEG system which would move with your head. So you could do all kinds of fancy things with it. And then in principle, if we could solve the problem of noise from the MRI system and have a sensor which is tolerant to high magnetic fields, we could measure MEG in the fMRI. And also we could measure MEG simultaneously with TMS, so that we could have not only multi-channel MEG system, but a multi-channel TMS system, which would allow us to focus the TMS stimulation to different parts of the brain in a very rapid temporal sequence, so that you could stimulate, for example, 2 areas of the brain in a very short delay with respect to each other. So, let me
TMS with MEG?
we have obtained funding to do it with atomic magnetometers, so that's why I have this picture here. And we have tried to use starting with 2 TMS coils simultaneously. So I hope this will become reality. So let me do another skip to the future and cop. Now, where is the? So I have gone through all kinds of properties of different methods, and it's hopefully become clear that there are both technical and physiological limitations in all of these methods. So you cannot hope that one method solves everything. And therefore you should seek for the questions first and then find the right tools to answer these questions. And some people are very dogmatic about this hypothesis-based approach, so that they almost deem it wrong to explore your data. I think you are still allowed to look at your data and modify your experiment according to your data and so forth. You cannot know all the hypotheses beforehand. And therefore I say that the Philosopher's Stone is not the method, really the imaging method, but it's the whole approach. It's the experiment, it's the tools that you are using, and it's the analysis methods that you are using that together make up the Philosopher's Stone. And my final comment is that don't be afraid to try something new. Professionals built the Titanic while amateurs built the Ark, and therefore we are brave to try something like fMRI and MEG combined and TMS and MEG combined. So thank you.